Welcome to today's lecture. Today I am going to talk about carbohydrate digestion. In my previous video, I told you about absorption of digested sugar from our small intestine. They were glucose, fructose and galactose. But how the carbohydrate we eat in our diet get converted into those forms? So from this it is quite clear that the importance of digestion is conversion of higher complex molecules into simpler form that can be absorbed in our blood circulation. So talking about carbohydrate digestion, major carbohydrates in our diet are starch, sucrose and lactose. So digestion of carbohydrates start from mouth. In mouth we have enzyme salivary amylase, also known as ptyalin. So this salivary amylase acts on starch to give maltose and other glucose polymers, around 2 to 9 polymers of glucose. But in our mouth we chew for a very short period of time, due to which only 5% of starch gets digested. Now the whole content goes into our stomach. Into a stomach we don't have any enzyme for such digestion, but this salivary amylase continues its activity over there until for 1 hour, after which gastric acid decreases its activity, and finally the enzyme becomes functionless in the acidic pH of stomach. So in stomach what happens is around 30% of starch gets converted to maltose plus 2 to 9 glucose polymers. Now in duodenum we have secretions from pancreas, which is pancreatic amylase. It is responsible for complete digestion of starch into maltose and glucose polymers. Now the content moves towards small intestine in jejunum, In jejunum, the secretions or the enzyme are present in jejunum. Enzyme are present in microvilli lining enterocytes. Enzymes present are sucrase, which will digest sucrose, lactase, that will digest lactose, maltase, that will digest maltose, and alpha dextrinase, that is responsible for digestion of dextrins. Now, I will talk about digestion in jejunum in a bit detail, which is important to understand. So here, now talking about, this one is an enterocyte from a small jejunum lining microvilli. This is membrane in which these enzymes are present as an integral protein. So our substrate, which is starch, starch was converted into maltose and glucose polymers. So we have lactase enzyme that will convert lactose into glucose plus galactose. This enzyme complex is sucrase isomaltase complex. Sucrase isomaltase complex. Sucrase part will digest sucrose into glucose plus fructose. Isomaltase will cleave isomaltose into glucose units. Now these isomaltose are similar to maltose, except the fact that isomaltose have alpha 1,6 linkage rather than alpha 1,4 linkage present in maltose. Now let's talk about another enzyme complex. This one is maltose glucoamylase complex. This enzyme complex will digest maltose into glucose units and then glucoamylase. So this one is maltase glucoamylase complex. So maltase part digested maltose into glucose units. Glucoamylase has the capacity to cleave both alpha 1,4 and alpha 1,6 linkage in dextrins and produce glucose. So what are dextrins? Dextrins are intermediate product of starch hydrolysis. It have both alpha 1,4 and alpha 1,6 linkage, which are broken by glucoamylase enzyme to produce glucose, which is final product of digestion. So in this video we saw how complex carbohydrate like sucrose, lactose and starch So in this video we saw how complex carbohydrate that we take in our diet, starch, sucrose and lactose, get converted to simpler and absorbable form like galactose, glucose and fructose, which are then ready for absorption in our blood circulation. So this was all about digestion of carbohydrate. Thank you for watching.